This is Deepak.
Okay.
And Whitney Graybill. I'm on the phone.
Okay. So that I know that this group has kind of a variety of experiences with the grant system. So and everything from people who have submitted grants who are doing revisions to, I think, people who are thinking about submitting their first real proposal. So some of the stuff I'm going to say might be like, yeah, I know that, and I apologize. But I guess if nothing else, what I want you to do at the end of the session is to think about what you're reviewing, who is your audience when you're doing an NIH grant review, because or grant writing. And the problem that people often have is that they have interesting ideas, they have problems that they really want to solve. They're after NIH to give them money to solve it, but they forget what that audience really is. And so the thing that, what's a significance statement? That's, I don't know how much you put on it. This is something that we'll talk about in a second. The thing that I actually want to talk about the most is what is the NIH Reviewer really, sorry, I thought I would have time. What is the NIH Reviewer really doing with your application? What have they been asked to do in relationship to the NIH? So the thing that you have to probably understand the most is that the reviewer of an NIH grant is not there as your mentor. Okay? The reviewer of the NIH grant has one job and one job alone, and that is to rank applications that they are asked to review in regard to their relative impact on the field. So when you're writing these applications, that you have to always keep that mission of your reviewer in mind. And where that comes from is that there was, and this was actually a huge change in NIH culture that came about 7 years ago in relationship to score. We used to be asked to rank the relative quality. We were allowed to, mentor applicants. Now it's just, we're asked to determine what the project's impact will be on the field. And the thing that you need to understand is impact and significance are in fact different. So the significance of a grant, not the significance that you're going to be writing in your application, but when a grant reviewer reviews you on significance, this is essentially a measure of, is this, say, an important problem? But it's not just that it's an important problem. I mean, if you're studying, let's say, glioblastoma, that's an important problem. People die of that. There's not a whole lot of cures. But just because you're studying glioblastoma does not mean your project is significant. Is your project significant to the field of glioblastoma? Is it something that, if the aims are successfully completed, is that going to move the field forward in some sort of interesting way? So you can have projects that studying the problem is highly important, but the significance won't be all that much because the aspect of the problem you have chosen to study is going to be judged by that study section member as something that is, it doesn't seem to be that relevant or that interesting in relationship to that problem. Okay? So, of course, you need to sell significance. We've always had to sell significance of our grants. Where this change in score came is what the actual score means. And the score that is the important score you have to worry about is called the impact factor. So the impact factor, or the impact number, is a scoring range from 1 to 9. whole numbers only. Now, impact is not just a measure of whether it's significant. It's a measure of not just what is the chances that it's actually going to accomplish what you're after accomplishing. So you can have the most significant project, but it will get a poor impact score because your approach is not convincing that it's actually going to go after the significant problem you're addressing. It might get a poor impact score because you're considered an investigator that is not competent to carry out the project. It might be that the environment doesn't convincingly have, since you're all clinicians, the patient load necessary to do the project in a way that is statistically significant. So if you only see 4 kids with this disease, that is, the impact's going to be bad because you're not showing you can actually accomplish what you are stating because you don't have that patient load. So when you're trying to write the application, in the end, you need to convince, honestly, 3 people that this project is not just important, it's going to get done if it's funded as proposed. In other words, with you as the principal investigator and with the environment as you have explained it to the reviewer, okay? And this is, and that also includes, do you have the patients necessary? Do you have a way to get patients to you and all of these aspects? So when there's I think another one of the sessions that we're talking about all of the ancillary information in regard to how to write an application. That stuff's really important in assessment of impact. So today I'm going to focus on trying to convince your reader that the project is significant as well as that it's feasible in the sense of approach. Okay. So is everybody good on that, like on the MUSC side?
Actually, Ryan Arnold joined us from Christiana Care.
Okay, and whoever is from Christiana Care, is that okay? All of the remote people good? Okay. I think you're muted if anybody's saying anything.
Yeah, I can hear you.
Okay. So if there's any questions, just interrupt me because I can't see you in this room. So don't hesitate to. I have this tendency to get into lecture mode, so I'm happy to have this be a conversation. It's just I'm used to more lecturing than conversations, but please let's make this a conversation. Okay. So the thing that we need to talk about then is how do you actually put together an application that is going to be convincing to the reviewer that it will meet these criteria. So one of the things that I sent Aaron a large number of like articles, and I'm just going to open up my laptop so I can pull them up here. Just so that I'll tell you briefly what they are, and then we'll get to what I kind of wanted to talk about today. So one of the things that's in there is, it's actually an example Ames page from a funded R01 that is actually active. It's actually my current R01. And I put it there as one of a number of example styles that I would absolutely go to all of your mentors and ask them for example Ames pages of funded grants. These are grants that you know have been successful through peer review. Okay. Now, from the standpoint of, there's also an article in there that's talking about making sure you get things done in enough advance so that you can actually get feedback from your mentors. And I think everybody knows this in their heart of hearts, that it's better to get your grant reviewed before it's really reviewed so you have a chance to fix it. But there's 2 tendencies. One really big one is, of course, you're rushing, you're all busy people, you get it done at the last minute right before the submission deadline. The other issue that people have is it can be made more difficult emotionally and psychologically to get critical feedback from your mentors, from the people who, you know, I guess in your case, your clinical super, you know, I don't know what it's called, but basically the boss ahead of you in the hierarchy. Or in my world, either, you know, professors in the department that will someday vote on your tenure or your faculty mentor. People don't want to look necessarily bad in front of these people. But the deal is that you want to get that feedback first. And there's an article in there about the psychology of that. There's also, I put in there, so there's a file called Impact Significance. That is talking about the NIH definitions of impact and significance. And it's actually a federal, it's a federal form that it's like a federal release talking about the official definitions. There is the critique template. In other words, this is what we are asked to fill out as NIH grant reviewers. There's also just a copy of the directions from SF 424, which is the research plan from NIH grants, and the instructions that reviewers are given when we're asked to do the critiques. The other set of articles are more professional development things. One is how well do you take constructive criticism, and how badly many people don't. Many people do. There's also an article on policing your prose. This is a very extreme article where they take one paragraph out of a physics paper and they get it down to one phrase by deleting a lot of excess verbiage. But that's actually really important. Very often when folks say, I just don't have room to discuss this important aspect, or you get your reviews back and they say, well, you didn't discuss this pitfall, and you didn't talk about that, and you didn't talk about that, and you feel there's no way there's room in the application. And it's true, it's hard to get all that into like a 12-page R01 application, or I think it's 6 pages now for an R03 or so. But often you can really make things concise. You can get rid of a lot of filler words. You can not say the same thing twice. And in grant writing, being concise but clear is critical, because if you can be clear, but if you're not concise, you're not going to have room to talk about everything that's an issue in the application. So it's something that you can read about. There's a couple articles on talking about how to interpret grant reviews, which I'll do a whole session on that later. But one of them, which is really relevant to this discussion, is they're just not that into you. Erin, is it possible I get a glass of water?
Yeah, I'm going to run to the cafeteria.
I'm okay, thanks.
So from the standpoint of they're just not that into you. So the thing to realize is that the reviewer is not required to mentor you. And very often people will read the application and they're like, nothing wrong with it, really. So it's just like, don't really care, it's not that interesting. But as a reviewer, it's really hard to write, this is just boring, or that this really doesn't float my boat, I'm going to give it a bad score. And the reviewer needs to look for, their feeling is this is a score of 4, but they need to write the review to justify that score of 4. So you'll get these picky details, like, oh, they didn't describe this scenario, or they didn't describe this or describe that, and you can get thrown into circles trying to address the reviewer comments, when the fundamental problem is that you didn't address the issue, is the project interesting? Is this something important that really excited who your reviewers would be? And then it sort of talks about that. Now, the other thing, this article, The 3 Deadly Sins of Grant Writing, is sort of interesting because it's talking about, actually, actually the article I'm thinking of is the funded menu. It basically makes an analogy of a grant versus a restaurant menu. Okay, so are you going to buy, if you have two restaurant menus, do you want the one that is fried chopped beef steak with coagulated cow protein, or do you want the artisan beef, lovingly raised on the ranch with Gruyere cheese or something like that? It's making this analogy of having to market your science, and a lot of this application is marketing. And there's also a couple books out there about marketing for scientists, and it's something to consider, to really think about grant writing as a marketing exercise to your reviewers. Okay. So when you are formulating your application, you want to think about this as your job is marketing your ideas to the reviewers, who are either going to support your marketing to the NIH to give it a good score, or they're going to say, I don't buy it, and then it's going to get a bad score. Because you have to get through the primary peer review. After that, there could be some vagaries, that if you don't survive primary peer review, you're never going to get funded. Now, one of the things that's really important when deciding how to market is you have to know who your audience is. Now, one thing about the National Institutes of Health, and particularly for young investigators, most of the time you have some ability to get information about who you're marketing your application to. Okay? So all of your grants are, unless you're going for a K award, the grants are going to go to the Center for Scientific Review. And there are basically panels that are called the permanent member panels. These are going to have lists of the names that are reviewing grants in certain topics. That you should absolutely, before you send in an NIH application, you should figure out what grant review panel is most likely to review your application. Different grant review panels have different scopes, and if you think that your grant straddles two scopes, absolutely look at the grant panel and read up on what everybody's scientific expertise is. Yeah.
Now, does each grant review, are the grant review panels exclusive to different institutes?
Okay, generally, it depends on the type of application. Pretty much uniformly, our awards are not inclusive to institutes. So whether it's an R03 and R15, those sorts of awards, the reviews of them are done by the Center for Scientific Review, which is an entity separate from any institute. Okay? Those are the Center for Scientific Review is what does all of the incorporated study section panels. Okay? So you might hear somebody say Dev 1 or Med 1, or my grants go to something called BVS, and then there's another one in my field called BVS. One's basic visual science, the other one is diseases of the visual system. Those panels are not run, in my case, by the National Eye Institute. They're run by the Center for Scientific Review. For those panels, the majority of grants that go to those two panels are, in fact, funded by the National Eye Institute. However, there's a lot of research on diabetic eye diseases. The majority of diabetic eye disease grants go to neither of those panels. They go to diabetes panels, where they're going to be reviewed side by side by grants that might be going to NIDDK, because the issues have to do with diabetes disease mechanisms. So they're going to diabetes disease mechanism study sections. Okay. So when you're considering, and you can do things to drive your application to a particular panel. It doesn't always work. But when you're going to apply, so you look at all of the panels, you decide which ones seem to fit your grant the best. That when you submit your application, you're asked to include a cover letter. That's the place where you suggest a study section panel. So you have that opportunity to say, let's say there's 3 panels where your work can go, but you look at who's on those panels and you're pretty sure that those folks, they might have heard of you. You might have talked to one of them at a conference, and they seem to think well of you. It could be that you are that you look at the panel and you know that this set of folks seems to have the right background so that they can understand your work and appreciate it. So under that circumstance, you can request that your grant is reviewed by a particular panel. And that can be very, very useful, because otherwise what happens is that your grant goes into a big, in the old days it used to go into a giant room, and there would be stacks of photocopied grants, and somebody would have to sit down and read all the abstracts and say, this looks good for that study section, that looks good for that. Now it's done electronically. But if you have a preference, you need to drive that preference. Now, it doesn't always work. They don't blindly say, this grant needs to go to this study section because you requested it. But you can definitely, if somebody reads it and it's like, I'm not sure between 3 study sections, you can break that tie. Okay? Now, sometimes after the grant is assigned to a study section, you'll get the initial study section assignment. The grant will go to a different study section, because once the review administrator starts looking at stuff, they realize, this is actually better here. And you can't really affect that or do much about it. But it's things that, it can help your case to get it into the study section you prefer. It doesn't always work, like I said. And the reason that you really want to have some idea what study section is, you need to know what the audience is, and you need to you want to go through the list of permanent members and at least know what their academic area is. So if you're studying something, now also keep in mind that your grant might not be reviewed by anybody on that list, or it might be reviewed by 1 or 2 people. They bring in people from the outside all the time as subject matter experts on the study sections. So that doesn't mean that you're definitely going to be reviewed by one of those names on the list. But it still gives you an idea of who you are writing this document towards. Is this a study section that is going to see a lot of clinicians? Is it going to see a lot of, I could imagine clinical psychologists could see your stuff. Yeah. And it could be that a clinical psychologist is going to look at this very differently than if you do obesity, right? If it goes to an obesity panel where people are studying leptin and hormonal signaling, they're both obesity projects, but depending on which study section it goes to, people are going to look at the work very differently. Okay, so does that kind of make some sense?
So just to reiterate, so you were saying that I just came online just to see if I can find it. So if we're thinking about submitting an R03 or R21, that's not institute specific, that's just an award?
For the most part, R21s are done by study sections. Now there can be exceptions. Sometimes R03s will go to an institute, but one thing that you could do is ask your institute's program officer. So if you have a program officer that you need to figure out who at your home institute administers the grant you're trying to apply to. Okay? So if there's any uncertainty about who's going to review it or anything like that, it's just pick up the phone. You can go to the institute. All the institutes have, these are the program directors who are in charge of these different aspects of their extramural program. And you find the person because that's the person who's going to make the final funding decision, right? Most of these folks are very interested in mentoring young investigators, because their job is to build a very, very important portfolio, right? So they want to bring young investigators in. It looks good to have young investigators be funded. Also, then they've heard your name, so that if you're on the line, they're like, yeah, he's a good guy, I'll fund him. There's human interaction involved in this too, so it doesn't hurt to ask for, like, let's say you see a RFA for a R03 application. There's usually going to be a contact information in that R03. Or if you just know that your institute funds R03s, on their website they will say who's in charge of what types of grants going to that institute.
You said each institute is one program officer?
No. So like, for instance, in the National Eye Institute, which is one of the smallest institutes, I believe there's 4. So there's one that does lens and cataract, one does retina, one does glaucoma, and then there's one that does kind of like training program type stuff. So, and that's the National Eye Institute. Cancer and IDDK, all of these are way larger institutes. They're going to have many more.
So I would be going through NIAMS, just so I find some musculoskeletal. I'm trying to find where on this particular institute, if you would just type in program officer.
Well, I would go to the institute homepage, go to extramural, and then there's usually about us or contact us or something, and then there's a whole list of all the program officers.
At that particular institute.
At that particular institute, yeah. Usually you get to it through the institute websites. And I can definitely help you try to find that later if you can't find it. Okay. Is everybody good so far? About audience. Okay. And knowing your audience is crucial here, because a good grant is written by getting into the head of who your audience is. Now, the first thing I want to kind of talk about is the aims page. That you should consider, while NIH grants make you include a lay abstract or a clinical significance statement and an actual abstract, that I have never met a single reviewer who reads any of that, okay? You have to include it, it's required, but it's not really written towards the audience of your reviewer. Those things might be written when they're trying to assign study sections, but usually not. It's usually the aims page. That's the public thing that Congress and everybody sees publicly if your grant is funded. The abstract is what's going to end up on the NIH website saying, I funded this grant doing this work. Abstracts are usually very vague because people don't want to publicly disclose what their research is. You know, like you'll say, this is what I'm studying, this is why it's important, but you're not going to say, I'm going to do these experiments in that abstract mostly. The clinical significance, that's there so that NIH goes to Congress and writes their reports, that they can say, you know, this it might be a basic science problem, but it has really big clinical significance, or you guys don't have this issue because you all work on clinical things. But that's, again, not there for, I'll put on my reviewer hat, for me as your grant reviewer. That stuff is all for regulatory stuff internal to NIH, okay? The thing that is the actual technical abstract that reviewers read and consider the abstract, to be honest, the program officers read and consider the abstract, that is the specific page, okay? And that is the document that will be your hook actually, Aaron, can you do me a favor and get my aims page up on this computer? So it said technical abstract. So when we're talking about putting together an aims page, we essentially want to make sure that the entire project and why we care about it is in fact encompassed in the in what's in that abstract. So from the standpoint of did I send you the file how to get funded? To send to them, I have a feeling.
Funding grant, recipe, that is not in that.
Yeah, I think I didn't send you the how to get funded one. Okay, so the how to get funded file is, that's the, actually the one, if you could pull it up, is the one that is the funded, aims page from funded grants. So I put this in here because this is an aims page from one of my grants. The first thing is got a big, colorful picture of some sort. Don't try to make a reviewer just read prose. Diagrams are really important as you go through a grant. The other thing here is that from the standpoint of a structure of an aims page, that the first thing is it sets up the problem, starting at the level I'm expecting my reviewer to understand. So, from the standpoint of the folks here, this is not enough introduction for you, right? Because you might have not thought about the eye or the lens since you were in medical school, right? But from the standpoint of my reviewers are going to be people who are definitely ocular biologists. They are likely to also be people who are very, very familiar and experts in this tissue. So I don't need to talk about the lenses doing its roles in light refraction. That I can basically start with my basic, this is the issue in this tissue that I'm studying. Because anybody reading this will understand it. And that's important because I wouldn't want to give so much intro that I couldn't get to the important bits of what I want to study. So I'm basically talking about, this is the big problem that or the big tissue or problem I'm interested in. It's then saying, why is it important? Why do we care about it? And it's important because it's involved in cell survival, differentiation, blah, 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 blah. And then it's saying that this molecule that I'm studying is one of its components. We don't know what it does, but we have preliminary information that says we figured it out. Okay. Then I'm basically talking about how is this protein detected. We also have some information saying that those detection molecules are also important. So I'm essentially laying out, this is the topic of the grant. This is what we don't know. This is what we think is going on, and we have evidence. That then takes us down to, again, we don't know how all of this ties together into this disease. So we have this aspect, I have this aspect. These two aspects tie together for this disease, and this disease is called posterior capsular calcification, which is what my grant is. Now, that says, okay, we don't know all these things. So then I'm going to come down to saying the objective of the grant is to figure out how all of this stuff crosstalks in regard to treating these diseases. Okay? So I'm again trying to, you can almost think of this as taking your reader by the nose. I know this is where you're starting. You know that this is an interesting thing. You probably already know we don't know a lot about this. But then it's saying, hey, look at me, I've kind of figured a part of it out. And it's then taking you down to what is the big picture thing that you're interested in. Now, sometimes this is done, so this is the way I did it in this application. Another way people will often do it is saying, we know this, we know that, all of this evidence together suggests that this is true, leading to a hypothesis. So that's the global hypothesis of the grant. Okay? There's reasons that I did not structure my grant this way, because my 3 aims are they're not all that interrelated. So they're interrelated in relationship to this whole understanding this tissue, but they're not as related in an overarching hypothesis. So in this case, the objective is what I put as my overarching theme of the application. For many of you, your overarching theme is going to be your hypothesis. Like if I remember reading your application, your hypothesis is going to be, can you intervene in helping these families deal with obesity by these video educational modules? Right? So that would be your stated hypothesis right in front. Now, we then have aims. Most grants have either 2 or 3 aims. And if you're doing like an RO3, RO3s are often on the 2 aim side. You're not obligated to do 3 aims, for sure. Most grants have at least 2, though. I just recently reviewed a grant, though, that had 9 aims. And it was a very odd grant to read, and it was by somebody who's a member of the National Academy of Science. So the whole application was very strange, but I guess it works for him, and I don't know how that particular application is going to do, but now in my case, I have formulated my specific aims into my sub-hypotheses. So I have said that my aim is to test the hypothesis, and this is my hypothesis. This aim is to test the hypothesis. So here I have 3 independent hypotheses. That can be, when you're structuring a grant, be really important, because there was somebody's reviews that I read that they kind of got in trouble a little bit in relationship to having these aims not have independent hypotheses. And where that ran into was that you don't want to have, I'm going to do this, and then using this information, I'm going to do that, and this information, I'm going to do that. And the reason is that well, if aim 1 fails, the entire application fails, and we're not going to get anything out of this, okay? And that's a major concern of reviewers. So generally, you want your specific aims to be relatively, they can be salvageable. If one aim dies, the other aims will salvage the importance of the application. Now that's really important when we're discussing impact, because it could be that if all of the aims are done, it's really high impact. But if aim 1 is even slightly risky and fails, the impact of the application becomes 0, or in NIH parlance 9, which means no impact. Okay. Does that kind of make some sense? You're pondering.
Oh no, I was just I was comparing. I brought a sample of grant that I had written. I was just comparing it to what you wrote there, and I was just sorry.
That's okay. Now, some people like to put a lot of methodology in aims. I personally don't. That I really feel that aims pages are more valuable in to really, this is marketing again. The aims pages are selling that you are going to go after interesting and important questions, and leave the description of the methodology for the main application. Because you want to use an aims page to really make your case. And it can be very, very good to explicitly state the impact, why you think, you tell them why the impact, why this application, if done, is going to be high impact to the field. Don't let them try to infer it. Now, it could be they won't buy your argument, right? But what can be really deadly is that, keep in mind that 3 people taking the same evidence that is unprocessed evidence can come to 3 very, very different conclusions. Particularly since your grant reviewer is going to be reviewing this at 2 o'clock in the morning, 2 days before the reviews are due, after a really long day, and they just, while you've been working on this and thinking about this for months or years, the chances of them being able to come to the same conclusions from the same information in that hour and a half of tired reading is that you've come to with all of this thinking and processing are actually, it's very, very risky. However, if you lay out what you've done, you then say that this is why this work is really, really important and needs to be funded, that you've made it easier for them. You have pre-primed the thought processes. Now, it might not, it actually may not work. It could be that, your reviewer has different opinions, or the evidence immediately makes other interpretations come to mind. But again, this is marketing, you're selling this as much as anything. And it really can help to smooth those grids from the standpoint of telling the reviewer what they should be thinking. Is everybody good on remote, in a remote world? probably FUD. Like I said, anybody in remote land, if you have any questions, just chime in. All right.
Thanks, Bill.
Huh? Okay. So now from the standpoint of so we have from the standpoint of an Ames page. Now, one of the things that you have to be very, very careful about is that your hypothesis, your aims, and your experimental approach remain congruent as you are developing your application. Okay? That very often you have this hypothesis, but one thing that people do all the time is that when they start getting into the weeds of saying how we're going to do the approach, they basically lose track of, is that experiment actually going to test that hypothesis? Is it actually going to test that hypothesis definitively or not? And so people get themselves into some trouble there. That, in the end, it's like, well, you have this very nice set of experiments, but that doesn't match up with the hypothesis. And you'll get a bad score because what you're planning on doing is not actually addressing your hypothesis. It could be addressing another perfectly important hypothesis, then you need to rewrite your hypothesis. So these are important things that's why also sometimes being able to have your application done a couple weeks ahead of time, then being able to put it aside for a few days, go back to it and say, does this experiment really address my hypothesis or not? It's really, really important. All right. Now from the standpoint of, just to go through the different pieces of the research plan. Now, the required pieces of the research plan are pieces A, B, and C. And A is the overall significance. That this is where you really elaborate on whether this is the big picture scientific question. This is not the place for an information vomit. It is not the place to say that this is a comprehensive literature review of the field. When they redid the NIH grant application 7 years ago, the comprehensive literature reviews of the field went right out the window. You don't have room. That you are basically saying to the person, this is why we really need to be studying this. This is why knowing this piece of information is critical. This is where there's this giant gap in knowledge that you better fund me or the field is going to be held back. This is where you're convincing the people of that. Right, so this is, again, it's really where you have to be aware of the audience. You are also really don't want to try to sell the specific significance of each specific game. Okay? Particularly a longer grant like an R01, people are often unlikely to read it in one sitting. Okay? If you give all of the significance of the entire application in this significance section for A, the next time they come back, they will, in fact, forget what you told them last time. So this section A, in my opinion, is way better done as very big picture. All right. And so this is one thing I recommend weighting the significance of each aim along with the preliminary data for the aim within the aim. Okay? There is no longer a separate preliminary data section in an application. And for that reason, it's actually generally better to read your preliminary data right when you need it to sell your approach. Okay? Because again, think human nature. Somebody's trying to read this in pieces over a day and a half. They're trying to put it together. They did not read it in a sitting. You front load too much information. They're going to forget what you told them. And that's actually something that grant writers are always saying, I did mention it, it was right here. But when the reviewer was writing the approach score, they were reading the approach section. They did not go back and reread the significance section. Okay? So that's why I recommend keeping the significance section big picture. Now, the second section is innovation. And people don't necessarily know what to do with innovation. The innovation section, it could be methodologies. It could be trying a intervention approach, like in your kind of work. It could be trying an intervention approach that has never been done with a certain patient population. It could be bringing a technology that has never been done. It could be even addressing a problem that no one has ever even tried to go at before. Innovation, trying to write towards an innovation score can be really difficult though, because you'll get 3 reviewers and you can get a wide disparity in innovation scores, because different reviewers will see innovation in very different lights. Some people only think about technology. Other people are looking at, is this an innovative way to answer a question? Even if the technology's known, it's never been applied in this way. Okay? Innovation sections are usually pretty short. From the standpoint of overall significance, I try to keep these to a page, maybe less in like an R01 application, which is 11 to 12 pages.
Each?
Huh?
The significance should be a page, the innovation should be
No, the innovation may be paragraph. Innovation sections I keep really short too. Where I spend all of my space is in the approach. And the reason I do that...
I'm sorry, so the significance should be a page total?
The significant at the most a page.
And then the innovation, the
Innovation
paragraph?
may be paragraph. I mean, this isn't an R01 application. If you're doing like an R03, which is 5 or 6 pages, maybe even shorter. And the reason is that I say this is that this is where you use the approach section to weave in really selling why I'm doing what I'm doing. Okay? Because when you read the directions, it says it's a description of what you will do to achieve your aims. But it also addresses data interpretation, data analysis. It is there evidence? Like with the reviewers reading it, you're saying, I'm going to do this experiment. Well, is there evidence that you as the investigator know how to do the experiment? Is there evidence to suggest that this might even be the right idea? Preliminary data. This approach section, in my opinion, and different people will tell you some different things, but in my opinion, it's the most effective to weave the preliminary data and show your preliminary data in the approach section, not in the significance section. Because again, the approach section is a major driver of your grant score, okay? That even though when you look at the template, that might not be real convincing to you as an applicant, I will tell you as a reviewer, most applications have competent investigators. Most applications are coming from environments that are competent. You don't get a whole lot from somebody's basement. The thing that discriminates between applications typically is significance and approach. Okay? And those are the 2 things you really have to sell home. The approach only makes sense if there's some evidence to suggest that your idea is correct. In other words, that preliminary data. So some things that you can really think about is that, you start the approach section with like in my case, I had a specific aim. That was to test a certain hypothesis. My approach is, I usually have a paragraph starting the approach, which again almost reiterates that opening paragraph of the specific. It sets up the big picture problem. It sets up the overall objective again.
I have to go, but
That's
thank
fine.
you so much.
No
This was
problem.
great, actually.
Okay, how much longer are we?
I think I'm going to be done in like 10 minutes.
Oh, okay.
I'm going to try to do this quickly because I know all you guys have to get going.
Sure.
That then, as you go to your approach, that you start out saying, all right, this is my hypothesis. Then after that, you set up what evidence do you have that that hypothesis is correct. That's where you give the evidence from the literature that that hypothesis is correct. That's where you show your preliminary data to say that, that this is the problem. We think it's this. We think that this hypothesis is correct for these reasons. But to prove this hypothesis, we basically need to do the following sub-aims. And then your first sub-aim might be to say, all right, I need to address this specific question, and there you get into the weeds of what you're doing.
Do you specifically write out like how you would approach each aim, like approach the aim one and then go on about that?
So what I usually do is just break that up into sub-aims. So there's a section that's called approach, kind of lay out, again, the scientific problem. Then you give some evidence for a big picture hypothesis. Then you can have a sub-aim that basically says that, all right, this big picture hypothesis, I need to, if this is true, point A has to be true. So sub-aim point 1 might be to say that in my world, a protein is involved in a certain function. Sub-aim 1 might be to say, to investigate, to test the hypothesis that a protein is found in this cell type. Because you can't do the function if it's not there, right? So then you write how you're going to do that experiment to show that that protein is present in that cell type. There's where you talk about, I have an antibody that works. I've maybe done it at one stage in development, and I can see that it's there. Now I need to see where it is in the entire set of development, or I need to see how it changes in a disease process or whatever. So it's there. So sub-aim 2 is, what is it doing functionally? Well, I have some evidence that there's this human mutation that, people who have mutations in this gene are messed up in that process. But I need to have an animal model, so characterizing an animal model. Sub-aim 3 might be then figuring out what's wrong with that animal model, and so forth. And when you're actually developing this, you provide the information the reviewer needs to evaluate the strength of your claim right when they need it. Don't try to give it ahead of time. So if you're claiming that, there's a human disease, there's a human condition, well, maybe give some of the details of that phenotype there in the approach. And then you're asking, do genotype-phenotype correlations with an animal? Well, you say, all right, this is the human, here's the mouse. I'm just going to test this in the mouse because this is the human, because I'm building an animal model. You don't just talk about the human phenotype in the significance section and don't bring that back up again in the approach section. You need to walk the reviewer through, not expecting them to remember what you told them 2 pages earlier. Does that kind of make sense?
Definitely.
So which means that you don't have the space to repeat everything. So you don't say a lot in the big picture significance, but you give the details. Like you might say that this mutation causes some clinical condition. But then when you're doing the comparison with the animal model, then you get into the weeds with the details of what's wrong with the person. And I'm proposing to compare that with the animal to develop my animal model. Does that kind of make some sense? All right, so then at that point, it's really, in this approach section, it's also really important that you tell the reviewer what's wrong with your approach. In other words, there's no experiment that doesn't have caveats, so pitfalls. So you really need to tell your audience that you know what those pitfalls are. You know that, yeah, if, like I've proposed to do an experiment with an antibody, the antibody, we know it works for these things, but no one's ever used it for this application before. So if you know that this antibody's never been used for that application, the reviewer probably knows too. So you have to say, well, because this antibody's not been used for this application, it's possible that it will not work. So if I can't use this antibody, what are my other options?
So you have to have a solution.
You need to propose a solution. The solution could be doing a different experiment or trying to get it answering the question in a different way. The solution could be that I'm going to go after a different reagent, or I'm going to create a new reagent de novo. But what's important is that you know what the pitfalls are. Like, let's say in a clinical project that you expect to see 100 patients a year coming through. You need 90 to make your clinical significance. Well, maybe that year you're only getting 80 at the most. Well, then how are you going to go find the other 10 patients you need to make your study successful? So what other clinical sites can you call up to maybe help your patient recruitment? That would be a pitfall that you're acknowledging. This is the solution. And this is how I'm going to solve it. You don't want to have the pitfall is not the same thing as saying, well, I might contaminate my PCR reaction, or I may screw up. I've actually read from new investigators' pitfall sections that basically said, well, I might screw up in this experiment, so I might have to repeat it. I mean, that's not a pitfall. A lot of pitfalls are things that are beyond your control. Or let's say you're making an animal model, and it turns out that you delete the gene from the animal, and you don't repeat the phenotype, or the animal dies. That's actually common. You'll have a human disease, you mutate a protein, causes a human disease. Easiest way to make the animal model is to delete the gene. You delete the gene and the animal dies. So then the alternate approach would be, well, you can go to this more technologically difficult thing where you actually directly model one of the known human mutations or something like that. So you will say that this could go wrong this way, but I am savvy enough that I can solve this problem. And from a new investigator, they want to know that the new investigator is not naive and can solve they can recognize their problems at the planning stages of their experiment, and they can solve their problems as they go on. So there's always this section where it's talking about pitfalls, potential problems, alternate strategies. You also want to talk about what is your expected result. So if I do this experiment and my hypothesis is right, this is what my readout will look like. This is what the experimental result will look like. You need to say that explicitly. Then you need to say, if I don't get that result, that it could mean this hypothesis is in fact correct. And I will go after proving that using these approaches. So again, you don't want the reviewers to think that you have your pet idea, and no matter what the data is, I'm going to keep my pet idea. You want to show that you realize that you get an experimental answer. That could be proving the hypothesis, disproving. If it looks like it's disproving, what other experiments do I need to do to confirm my hypothesis is wrong? Our hypotheses are wrong sometimes. And the reviewers really want to know that you know that you could be wrong and that you're open to alternate interpretations of the data. Okay. Now, so that's really, really critical. At the end of each aim, you should reiterate why we care. That it all comes down to the reviewer's score is driven by why we care. So after you complete your aim, at the end of that aim, you never end on talking about a pitfall. You always end on that when this aim is complete, we're going to know this new, really important piece of information that's going to move the field along in whatever way it's going to move the field along. It's an important point.
Is it okay to only have 2, you said?
It's okay to only have 2 for a short application. You don't have to have 3. There's no obligation to have 3. And I've actually read 1 aim applications, to be perfectly honest. But most people have at least 2. 4 gets to be a little much.
Right.
So 2 to 3 is the sweet spot. But I actually submitted a grant recently that only has 2. But each aim has a lot of sub-aims. So it kind of depends on how do you want to break things up in relationship to how you do your project. Now, the other thing is that you will do the same thing with every aim you have. Again, you want the very, very last thing that the reviewer reads to be a sales document. So that you want them to leave your application saying you've just told them why you think they should be extremely excited about your work, so that they're giving you ones and twos. That you don't want the last thing a tired reviewer reads is, oh God, this might not work as a pitfall. You want to remind them that this is important and this is significant. So that, and this is that impact statement. Okay? So I basically say each aim in the entire approach section should have separate impact statements. So you're going to have a global impact statement up in your aim, up on your specific aims page. At each end of each aim, you're going to say this is the impact of this aim, and then you reiterate and tell them again that this entire application has a high importance of impact, giving you a good score. That way, but that kind of makes some sense. That is there anybody still remote? I went over a little bit, largely because I was late. Any questions remotely?
No.
No? Okay. So,
No, that's great.
Okay. So if it all makes sense for this aspect, I could keep talking about this for a long time, but I know everybody has things to do and whatnot. So that's sort of a overview in the sense, but I think the big take-home thing I want to say here, there are different ways to put together effective grant applications. One of the best ways you can get ideas of how this is done is to read as many funded applications in your area as senior investigators will give you, okay? You will see by doing this that there's different ways to accomplish the same goals. But in the end, the most important thing is you have to get out of your own head. You have to realize that you are marketing your science. And a lot of scientists find this really distasteful as a way to think about it. But if you don't sell your work, to expect you to just put it all out as a smorgasbord and that they're going to basically make the same decisions you've made is extraordinarily risky. It can work, but very often it doesn't, and then you're in trouble. So you need to drive the thought processes of your review.
Do you need a statistical analysis section?
Okay, so that is usually woven into the approach. I often in my applications have a statistical analysis of data section where I really kind of talk about my statistical support. But your statistics, like for instance, like in your approach section, you might say, I'm going to recruit 100 patients. Well, you better, in that approach section, explain how you did your power analysis to justify the recruiting of that 100 patients. Now, some of that also goes into your human subject section in the sense of where you're talking about your design. How do you came to, like in my case, I do a lot of animal work. How did I come to the conclusion that this is the appropriate number of animals to generate statistically significant data? So some of that data analysis and patient recruitment all goes into your human subject section. But in your approach section, you need to really convince them that, that I'm going to do this and we're going to do a two-tailed ANOVA with Bonnefoy correction or something. And you need to explain it enough to say why. My field doesn't tend to be as really sticklery about statistics as clinical research is. And a lot of that has to do with that most of my work is either, a t-test or a ANOVA. We almost never have to worry about, and we also, power analysis tends not to be so important because my animals are genetically uniform, so I don't have to worry about population-based genetic differences. Also, all my animals live in the same environment from each other, so I don't have to worry about environmental differences.
You use rats?
I use mice, but they diff. And so I don't have anywhere near the need to be very, very careful convincing my reviewers that I have enough critters recruited to be able to come to significant conclusions. You, as clinical scientists, that is big. You know, of convincing the reviewers that you have the patience that you need to do your study and make it meaningful is really, really important. I've seen a lot of clinical-based grants go down the tubes that way. So that's why, you know, that's why the CTR is, you know, really hammers on all you guys about statistical support and statistical consultations and all of that. Because if you don't have the statistics right, you don't have your N right, and then you haven't figured out that you have enough patience to meet that likelihood of like 90% confidence of being able to test the hypothesis, you know, you're going to be in big trouble. Any other kind of questions right now?
Do you have any thoughts on references? Which sections they should be in? Is it okay to have them in all the different sections?
Yeah,
How many you should have?
in my philosophy on references is I am hyper-thorough referencer. Okay? Personally, in the application, in my opinion, every single fact should be referenced. And that includes the specific aims page, that's the significance approach, all of the main body of the research plan, anything that you're talking about in an animal section or a human subject section. Because the references actually go at the end of all that. All of that stuff should be referenced. The front stuff, like the official abstract of the application or the official clinical significance, that doesn't need referencing. But everything from specific aims on, between specific aims and references, should be fully cited. And there is EndNote has an NIH referencing format that is probably a good one to use because now NIH wants you to include the PCMIDs for all of the references at the end of the application. It's actually buried in the directions. It's actually, if you use EndNote, it will automatically harvest it from PubMed. If you do it by hand, then you need to go spend $150 and get EndNote, or you're going to make yourself crazy. This is absolutely a situation where you want to spend the time learning the technology. And the reason I say this is that as you're writing the application, it can be really useful to put your references in line, like to say, you know, this is my fact, Duncan et al. In 2005, because you'll know what that paper is. You don't want to spend all that space in the final application. But while you're writing, you want to be able to easily say, oh, that reference goes with that. And you'll be able to tell. But you want to be able to change referencing formats on the fly. And this will also make you way less crazy when you go to write your publications. Because let's say you send a paper into a journal, it gets bounced, and you want to send it to the next journal. If you do it by hand, it can take you hours. Because reference 1 is, you know, it's your name et al. Whatever, while this other journal requires a superscripted little number.
Or some people like to list them as they appear in the manuscript as opposed to alphabetical.
Exactly, and well, and that's some things you can do, but some journals have requirements.
Oh yeah.
And so if you use EndNote, it takes a grand total of 25 seconds to change all the referencing format in the entire document. So I would absolutely say to you that if you're not using EndNote or an equivalent referencing software, and I would recommend EndNote. Which word processor do you use? Okay, so EndNote, it can plug into Word. So if you install Word and then install EndNote on top of it, it will put in a macro EndNote. It'll give you an extra little thing in your Microsoft Word, and you can work with that. It can go and can harvest all your references out of PubMed.
See, the problem is we're a little spoiled here, Aaron. I don't know if you know Dustin and Michelle, the editorial. So you submit the manuscript to them, and then they do the references for you. And if you want to submit it to another journal, they'll just do it for you. But I don't know if they do the same thing for grants. I've never really asked them to do that.
Yeah, something we can look into. But it's also important to keep in mind with them. I know they're getting their services are becoming more and more well known.
Right.
So the time that it may take them to do this.
It's longer. I mean, I have about 5 manuscripts in the queue with them, just me personally.
Right. I mean, and so that's the thing, is that there's certain things that, like for instance, for the NIH grants, like when I write my grant, I'll have it like Duncan et al. 2014, and then as I'm getting close to trying to figure out how long my grant is and stuff, I'll then switch it to NIH format and note. That will change all of my references to superscripted numbers. And then it will give me a numbered list at the end in the correct format. And like NIH, you have to have every single author name. They require at the end of
In
the
the references.
In the references.
You don't do 3 in it at all.
No, that actually I actually messed up a little bit in my endnote, so I got 4, then it went et al. And I got a reviewer who chewed me out on NIH grant review. I bet it was their paper that their name was left off the et al. But they chewed me out because I didn't follow. Huh?
Because you didn't list all
I
the
didn't,
authors in a reference.
I didn't list the Phillips. It wasn't compliant. NIH could have bounced my application without review because it wasn't compliant. And they're getting more sticklery about compliance now.
Yeah.
Now all of the papers have to have, in the reference list, have to have either PMIDs or PCMIDs. Where you can find those is in the PubMed record.
So you have to like type them in and get them.
Or you can just use EndNote and it harvests them out of the PubMed record and automatically stick them
Maybe have that, and maybe.
Dustin and Michelle, you mean?
Yeah.
Yeah.
there. I have my EndNote library is basically my 4th version of EndNote now, and I've been using it for 15 years. It has 3,900 references in it.
Oh yeah,
Yeah.
they save them, right?
Yeah, it saves it. So you can go back and, if you're in a field, there's certain papers you reference all the time, and they're just there. That I just submitted a grant a couple of days ago, and there was some new stuff, and I imported the new references into my thing, so I went from 3,900 to 4,000, and that's how I reference my new grant.
How many grants do you think people need to submit before they get one?
Quality is certainly better than quantity. There's no question.
I meant just like in terms of odds,
Well, some of it depends on what is your grant mechanism, right? I mean, because it's not all about odds. In my career, I probably have for NIH about a 65 to 70% hit rate. For National Science Foundation, I have a 0% hit rate. I have never gotten an NSF grant that I have submitted. I think I know the reasons for that, because the stuff I do tends to be the significance tends to be clinical. And when I try to write the same stuff, basic science, the basic scientists aren't as thrilled.
Right.
But, it's also, I've kind of been good at that marketing thing. There's some people who can write 30 grants and never get one. And some of that has to do with their sloppy applications. They're written in a dense way that people can't read them or understand them. They're working on problems that the reviewers don't care about. Or in one case, there's this one guy who just doesn't have a good reputation. His work is considered very, very sloppy. And, it really, makes it harder for him to get funded.
So there's no magic number.
There's no magic number. I am personally extremely fortunate in that I got my very first NIH application. My very first NIH grant I ever submitted was an R01, and I got it my first try. So I was very, very fortunate as a young investigator. But some of that was also planned. Like I really put a lot of effort in trying to understand how to put together an effective application. The resources there was a book that was out when I was a postdoc that was basically how to write an NIH grant and got funded. And I sat there and used it as my Bible. I did exactly what it said. you know, and it's no longer valid because the format's different. So that's why I don't suggest it to people. I used to lend it to people all the time, but it's really out of date. But, you know, some of it is not just sitting there and writing, but really, what's the word I want? Really understanding what the document is intended to convey. All of that said, you know, I had a streak that went from 2012 to 20, I guess it was 2012, 2013 is when this was really happening, where I submitted 5 grants and none of them were funded. And it's just like I couldn't seem to get past, you know, they were all scored, but I couldn't get below the pay line. And my research program had no funding for a while. And then I got one, but I got it under the pay line, you know. And same grant writer, doing the same kind of stuff I've been doing my whole career, but for whatever reason, for that panel, for what I was doing, you know, I had a streak where I had five grants. I was submitting every single cycle, 5 cycles in a row. I didn't get one.
Right.
You know.
But this is your career. This
Yeah.
is what you do, right?
Yeah,
so
this
like
is what I
someone
do.
like me, you know, I've been here 14 hours today,
Yeah, I
and
know.
I cram in an hour and a half of like grant education of my own personal time. But my point is, I've submitted maybe 4 or 5 grants to different orthopedic organizations, and none of them have been successful.
But are you getting feedback?
Oh no, yeah, I have feedback, and I've been submitted to the same project, and I keep kind of tweaking it. And I have another one that's out there that I'm